In artificial hybridization, it is most popular method nowadays. Okay, it is used to improve quality of plant. Okay, then it is used in plant breeding. Okay, that is crossing of plants. That means so that desired characters of two plants can be combined. Okay, that means this is one plant plus this is another plant. Okay, to combine both desired characters from first plant. Okay, this is one plant, this is another plant, and we form or produce or for combining these two desired, that is good quality plant. Okay, this is done by artificial hybridization. For this process, some steps are involved. In that, first step is selection of plant, and this process is done by manually. That means, for example, in sugarcane, first plant having character like long stem. Okay, then another plants have thick stem. Combining these two characters, we can produce a good quality, that is sugarcane with long stem and thick stem. Okay, and this selection is done by manually. Then next is, in one plant only female gamete used. This only from one plant female gamete is used, and from another plant male gamete is used. Okay, and if female gamete is used, then from this flower anthers, okay, is removed. Okay, anther is removed, and this process of removal of anther is known as emasculation. And for that, flower must be bisexual. If flower is unisexual, then emasculation is not necessary. Okay, then this removal of anthers is done by two types. First is manually it is removed, or if pollens are very small or powdery form, then suction pumps is used for emasculation. Okay, then next step is bagging. After emasculation, the female part, that is pistil, is covered with bag for prevent natural pollination. Okay, that means pollen grains come on stigma naturally, and if that done, then fertilization takes place, and for preventing this, bagging takes place. Then after bagging, collection of desired pollen grains. Next step is collection of desired pollen grains. Okay, select plant and cover flower with bag and collect anther. That means male plant or male flower part, that is anthers are collected, bagging for that flower. And after collection of anthers, next step is dusting of these pollens or dusting of desired pollens on stigma of emasculated plant. Okay, that is emasculated flower or stigma. Okay, then next step, fertilization. It takes place naturally. Fertilization and seed formation or seed formation. It is natural process. Then after seed formation, the most important step is testing of seeds. Testing of seeds. We test those seeds after germination. If new plant has long and thick stem, then our process is or we are succeeded in artificial hybridization. Then tested plant used commercially. Okay, this is process of artificial hybridization. And in our this chapter, last point is apomixis. Apomixis. Okay, it is type of asexual reproduction. Apomixis is type of asexual reproduction. Okay, that is asexual reproduction by agamospermy. Asexual reproduction by agamospermy means formation of embryo from an unfertilized ovule. That is formation of embryo from an unfertilized ovule is called apomixis. Okay, apomixis consists 3 subtypes. Okay, it again divided into 3 subtypes. First is adventative polyembryony means many nucleus fertilized, so called polyembryony. Poly means many. Okay, embryo develop from diploid cells. Embryo develop from diploid cells of nucellus or integument. This is called adventative polyembryony. Example is citrus plant or orange. Okay, then second type is recurrent agamospermy. Okay, embryo is developed from diploid MMC, that is megaspore mother cell. Okay, embryo developed from diploid MMC, that is megaspore mother cell. Okay, example taraxacum. Okay, and third subtype is non-recurrent agamospermy. Okay, in this type embryo develop from haploid unfertilized egg. Okay, embryo develop from unfertilized haploid egg. Okay, this process is also called parthenogenesis. Okay, this process is also called, that is non-recurrent agamospermy is also called parthenogenesis. Okay, in this type MMC, that is megaspore mother cell, undergo meiotic division, that is meiosis takes place, and meiosis is reductional cell division. That means from diploid mother cell megaspore mother cell, haploid egg is formed. Okay, and from that egg embryo sac, hence parthenogenesis occur and embryo is developed. Example is Nicotiana. Okay, in this way, first chapter, that is reproduction in lower and higher plants, here finish.